And now, yeah.
Okay. So shall I start?
Introduce to the audience. Okay. Okay. So good morning, everybody here in India, and good evening, Barak in Wisconsin. Ladies and gentlemen, today Dr. Barak Blum is our next speaker is Dr. Barak Blum. He is an associate professor in the Department of Cell and Regenerative Biology at the University of Wisconsin, Madison. Dr. Blum has earned his master's and PhD degree from the Hebrew University. Specifically, he studied tissue-specific transcriptional regulation of metabolic genes and tumorigenicity and clonality of in vivo differentiation of human embryonic stem cells under Professor Leah Reshef and Professor Nissim Ben-Ministry. He then did his postdoctoral research under Dr. Douglas Hamilton at Harvard, where he studied functional beta cell maturation in mice. Currently, his laboratory at Wisconsin combines genetic mouse models of diabetes, functional genomics, bioinformatics, and human pluripotent stem cell differentiation platforms to tackle questions related to functional maturation of insulin-secreting beta cells in the pancreas and cellular and molecular organogenesis of the islets of Langerhans, with the aim of understanding and eventually curing diabetes. Ladies and gentlemen, today Barak is going to talk on pancreatic development and stem cells. Please welcome Dr. Barak Blum. And Barak, it's yours now.
Thank you, everyone, and good to see you, Dr. Dara. Just the first question before I start, by show of hands, how many in this audience in the room that I see are undergrad students, undergraduates?
There are two only.
Two only, and the rest are graduate students?
Graduate or at the master's level? At the
graduate to master's level. Okay, very good. Thank you. So I will talk, as I said, about pancreatic, as Dr. Dara said, about pancreatic development and stem cells. Please stop me, raise your hand if you want to ask a question in between. I'll try to make time for that between sections. Wait a second. My talk will be divided into 3.5 sections. At the first section, I will explain a little bit about the biology of the endocrine pancreas, what are beta cells that make insulin, and about normal glucose homeostasis. Then I will explain a little bit about the different types of diabetes mellitus that we will try to tackle with regenerative approaches or stem cells. I will then speak about current developments and prior developments in regenerative approaches to treat diabetes. And at the end, if I have some time left, I will try to skim a little bit about stem cell-derived beta cells, aside from medicine or regenerative approaches and how they use in disease modeling and drug discovery. So the whole aspect, the whole venue of the talk happens inside the pancreas. The pancreas is an organ that in human sits just under the liver in front of the stomach. It's about the size, in human, it's about the size of and shape of a big banana. And depending on how fat it is, it actually has the colors of the ripe banana sometimes. And the pancreas is connected to the gut through the ampulla obiter that connects it to, that helps it secrete digestive enzyme into the gut. Now, the pancreas is really interesting because it's really a two-in-one organ. About 90-something% of the pancreas is devoted to secreting digestive enzymes into the gut. So these are these yellow corn kernels that you see on a cob. They are filled with granules with all the trypsins and the chymotrypsin and everything you need to digest your food. And these enzymes are secreted externally into the pancreatic duct and go to the gut, where they help us digest our food. But about 2% of the pancreas is called the endocrine pancreas, is composed of what's called the islets of Langerhans, which are these small red clusters in here. Let me draw them to you. They are the small red clusters in here. Second, do another color. There are these purple ones, and they are clusters of about 500 to 1,000 cells, some few thousand cells that disperse around the islet. Think about a bread with raisins. So all the acinar tissue is the bread, and the islets of Langerhans are the raisins. You see there's many of them in here. We have about few thousands, about 1 million in human. I'll show it later. And these cells are responsible for glucose metabolism, for regulating our blood sugar. As you know, most of the currency of energy that we use from food is in the form of glucose. And what the islets know to do is they know how to regulate the blood glucose in our body by secreting the mainly two hormones, insulin and glucagon, and a couple of few other hormones. Just a second. Here you can see a cross-section of these islets of Langerhans. I don't know why I don't have this thingy here. I'll erase it. Here you can see a cross-section of a islet of Langerhans. This is a 3D islet. This is a 2D islet. The red cells are cells called beta cells that secrete the hormone insulin. The green cells are cells that are called alpha cells that secrete the hormone glucagon. And these two hormones together are the most important hormones to regulate blood glucose. There are also other cells in the islets of Langerhans, mainly delta cells that secrete somatostatin, and more rare cells, pancreatic polypeptide, PP cells, and epsilon cells that make ghrelin. We think that these three cell types are less hormones and more paracrine interactions within the islets that fine-tune the expression or the regulation of insulin secretion and glucagon secretion. Now, islets of Langerhans are very conserved among all vertebrate. So about the size and the shape of islets is pretty much the same. The difference between animals of different sizes is not how the islet look, but how many islets they have. So in human, we have about 1 million islets. They have more alpha cells than in mouse, and less beta cells. They're about 50-50. The reason is probably the difference in what we eat and how we eat that determines how many alpha cells and how many beta cells we will have. Mice have about 1,000 islets, and zebrafish are born with one islet of Langerhans, called the primary islets, and then they have some tiny islets when they grow up if they need it. Now, the way islets or beta cells are working to regulate our blood glucose is determined by, or is due to some of their very, very unique characterization or characteristics that they have in them that enable them to regulate glucose. One of these very unique characterization is the ability of beta cells to sense the amount of glucose that we have in the blood and release just the right amount of insulin to any given concentration of glucose. And this is important because, and I will touch on that later, if beta cells secrete too little insulin to regulate the level of glucose in the blood, the level of glucose will be high and we will have diabetes. However, if beta cells secrete too much insulin in response to glucose, the glucose level will go too low, and that will cause it's a very deadly or dangerous condition called hypoglycemia. We can actually die from too low glucose. The way beta cells, and I will only do it very, very briefly, the way beta cells can do this is that they have a mechanism called glucose-stimulated insulin secretion, in way that beta cells are packed with granules or vesicles of insulin, and they behave pretty much like neurons do, except instead of getting neural signaling to release their vesicles, they get metabolic signaling in the form of glucose. So beta cells have a very sensitive glucose transporter that senses how much glucose is in the blood. Glucose goes into the beta cells. The more glucose you have in the blood, the more glucose will go into the beta cells. Glucose is metabolized. That changes the ATP-ADP ratio in the blood, in the beta cell, and closes potassium voltage-gated potassium channels, which leads to membrane depolarization. This is actually action potential very much like neurons have. And that action potential opens voltage-gated calcium channels. Calcium will get in the cell and cause the release of vesicles. This is pretty much like neurons do that, except instead of signals from the axon, what they get is regulation of blood glucose. So all the beta cells need to work is to be exposed to the bloodstream to sense how much glucose there is there. And I will touch on that later when we talk about using stem cells to treat diabetes. So how does insulin work in the body? What insulin does is that insulin is secreted from the pancreas. It goes into all cells of the body, and it binds to an insulin receptor. And what that does, it triggers the opening of glucose transporters that are on other cell types. They are different than the glucose transporter that's on beta cells. And they're related to, they are dependent on the binding of insulin. And the second insulin binds, these glucose transporters are open and glucose gets in the cell. So if you have high glucose, beta cells secrete a lot of insulin. Insulin binds to the glucose transporter. Glucose channels called GLUT4 open in the cell, and glucose gets in the cell. Blood glucose in the blood go down, and the cell gets glucose so it can use it for energy. Now, in normal glucose cycle, say you woke up in the morning, you just had breakfast, your blood glucose is high. As I said, this is just an illustration of what I just said before. Beta cells in the pancreas detect that there's high blood glucose. They secrete insulin. Insulin go into all over your body and opens glucose transporters. Glucose gets in the cells and lowers the levels of glucose in the blood, and the blood glucose are normal, and then we can live. If you have woke up late today and you're late for class and you didn't have time to have breakfast, the alpha cells in the pancreas will start to measure, to maintain your blood glucose at a normal range. That happens, so you woke up late, your blood glucose is low, you didn't have time to eat your breakfast. Alpha cells sense that your blood glucose is low. I won't touch how they do it. It's a pretty complicated process, actually. They release glucagon. Glucagon goes to the liver. The liver is the source of all excess glucose that we have in the blood. The glucose is stored in the liver in the form of a polymer of glucose called glycogen. And glucagon from the alpha cells triggers the breakage or release of breakage of glycogen in the liver in a gluconeogenesis, and breakage of glycogen that replenishes, that goes out in the blood, replenishes glucose in the blood, and your blood glucose goes to normal. Now, what happened when this process goes wrong? When this process goes wrong, we have diabetes. And as people here probably know, there are two main types of diabetes. The first one, which is actually the smaller type, the smaller percentage of people suffer from it is type 1 diabetes, which is an autoimmune disease. The other type, which the majority of population suffer from, is type 2 diabetes. Does anyone in the class here knows the difference between type 1 diabetes and type 2 diabetes? If you know, raise your hand. Don't be shy.
Question, whether do you know the difference between type 1 diabetes and type 2 diabetes? If you know, then you know.
Yes.
Hi, hi, sir. Hi, sir. Good morning. My name is Prabhanjan Das. In type 1 of diabetes, due to destruction of a beta cell do not produce enough amount of insulin. In type 2 diabetes, there is some destruction in the beta cell receptor to do not utilize the insulin.
This is this is almost close. This is actually very close. Very, very good. Excellent. So type 1 diabetes is an autoimmune disease in which the immune cells kill beta cells. Type 2 diabetes is a disease in which beta cells at the beginning can make insulin, but the body does not respond to insulin. It's called insulin resistance. So in type 1 diabetes, what we have is that the immune system somehow recognizes the beta cells in the body as a foreign entity and attack it and kill it. What you can see here at the bottom are actually pictures from my lab when I started the lab and still worked on type 1 diabetes. You can see an islet of Langerhans stained for red with insulin, and you can see immune cells stained here for purple for CD45. These are T cells. And you can see through the progression of the disease how the immune cells recognize the islet and surround it, and then start just breaking it apart and eating it. And a few weeks later, we have a scar pretty much that has only immune cells and almost no beta cells survive. And what that causes is that once all beta cells are gone from the pancreas, we don't have any other source of insulin in the body to use. So we eat, glucose goes up in the blood, but there's no insulin to be secreted from beta cells. Mainly, there's no beta cells to be secreted to secrete the insulin. Insulin can't bind to the insulin receptors on the target cells of all the body, and glucose can't get in the cell and stays in the blood. Now, this actually causes a phenotype that is more severe than just having high glucose in the blood, because glucose is the main source of energy all cells use to survive. So when type 1 diabetes, before the invention of recombinant insulin, I will touch on that in a second, was the terminal disease, was a deadly disease, and people would actually die from symptoms that are very close to starvation. They were literally starved because they would eat, their food will become become glucose in their blood, and this glucose would not be able to get into the cells and use an energy, and the people would just literally starve from lack of energy. Type 2 diabetes also causes or also is characterized by high glucose in the blood, but the causes are different. So what happens in type 2 diabetes is that we do have beta cells, they can secrete insulin, but for some reasons we don't fully understand, the insulin receptor either stopped recognizing or binding insulin, or it can bind insulin, but it doesn't transmit the signaling down into the cell, and the glucose transporters in all our target cells stay closed. Glucose cannot get in the cell, and we have a condition in which beta cells make a ton of insulin, but keep on feeling that the glucose remains high in the blood, because cells cannot translate this excess of insulin to let glucose get into the blood. Eventually, beta cells will work hard and hard and hard, because they will try to make more insulin and compensate for the glucose that doesn't go down enough. They will work. In most people, these beta cells will compensate by making more beta cells and making more insulin. But if the patient doesn't resolve the doesn't resolve the glucose or the insulin resistance, then these beta cells will overwork themselves and start dying or dedifferentiating. And then we just lose the source of insulin, the beta cells, and we get to a condition that we call type 1-like type 2 diabetes, in which this is the condition where this is a stage of type 2, that stage of type 2 diabetes where people that you hear about start receiving insulin. This is mainly because their beta cells have overworked, secreted too much insulin, worked themselves to death, and then you don't have enough insulin. So you have to get insulin, and you have to get more and more of it because your body is resistant to insulin. We don't exactly know the reasons for someone developing insulin resistance. There are definitely the clearest evidence that we have is for nutrition and exercise, so lack of exercise and increased body fat will be definitely a cause, but there are also some really cool and complex genetics behind it. So some people can be very obese but metabolically healthy because they will grow and grow in obesity, but their insulin resistance will remain mild, or their beta cells can compensate forever. Some people have beta cells can just really, really compensate well and keep on their glucose, secreting more insulin to mitigate this elevation of need in insulin. And these people can be obese and very healthy. In contrast, someone can be very lean and eat one extra dosa in the morning, and they will flip. The beta cells will just work too hard and will not be able to compensate for the increased demand for glucose, and the person will get type 2 diabetes. There are also other types of diabetes that are more rare. One is gestational diabetes. That is actually an interesting condition because a person that is pregnant needs to elevate their insulin, the glucose levels in the blood just a little bit so the embryo get more glucose. And in a person like that, if beta cells can transiently compensate, they will have gestational diabetes that will be resolved after they give birth because their body doesn't need to have glucose enough for 2 people. So they kind of resolve the transient insulin resistance. However, if a person gets gestational diabetes, we know usually that they're an increased risk for type 2 diabetes because later in life, we know that their beta cells may have a problem in compensating for lower threshold of glucose. There are also diabetes type 3C. This is a new type of diabetes that's been diagnosed kind of recently in the last several years. This is diabetes that's related to inflammation of the pancreas, what's called pancreatitis, or to cancer in the pancreas. We don't know exactly what it causes, but we think overall inflammation, assault of the pancreas due to cancer or due to other types of pancreatitis can cause beta cells not to work well, and we have type 3 diabetes. And there's also a very rare condition called mature onset diabetes of the young, MODY, which is caused from usually a mutation in one gene that has dominance in the activity of beta cells. There are 14 MODY genes. I had room here for 12. I didn't put all 14. But most of the MODY genes of mature onset diabetes of the young are genes that are related to beta cell function. PDX1, glucokinase is a gene that phosphorylates glucose when it gets in the cell and keeps it in the cell. A lot of those are transcription factor, HNF1A, PDX1, HNF1B, NORD1 are genes that are related or important for beta cell function. Insulin, of course, mutation of insulin causes diabetes. But some of them are really interesting, like MODY8, which is caused due to mutation in a gene which is active in the acinar cells around the islets. We still don't know why an acinar gene will cause diabetes, and it's a really interesting area of research. Anyway, either type of diabetes, if the diabetes progresses for long enough and all beta cells are lost, the person doesn't have any other cells in the body that can make insulin response efficiently to blood glucose. And what happens is that the patient, before insulin was discovered and about 102 years ago, exactly, in Canada, patient would die from starvation when they had type 1 diabetes. Just they did not have a source of insulin and they will die. With the invention, with the discovery of insulin and the invention of recombinant insulin that can be used externally with shots, the disease became from a terminal disease to a manageable disease. People prick their fingers many times a day or have now glucose, continuous glucose monitoring. These are glucose monitors that are implanted under the skin that sense the glucose and tell them every time they need insulin, the people will inject the bolus of insulin. Now, approximately 10% of the world population is affected with diabetes, and about 35%, so about 1 in 3 of us in the room here is going to have pre-diabetes or is close to becoming diabetes. A lot of that is because our food we don't exercise enough as we used to in previous generations, and we eat a lot of processed food. So this is an astonishing number of, I think, of people that are having pre-diabetes in the world. It's about 500 million people actually have diabetes. Now, the injection of insulin can can manage the disease, but it can't really cure it. You can see here the blood glucose of a person with type 1 diabetes with and without exogenous insulin over the course of 24 hours. The blue bar here is the maximum or the high, what we could consider a high level of glucose in the blood. The green line here is the normal range that blood should be, between 70 or 80 milligram per deciliter. Blood glucose to 120, that's a normal human blood glucose. 180 is very high. And then we have glucose that's below 50 milligram per deciliter, which is called hypoglycemia, and is a very dangerous condition where people can actually faint and sometimes die. And you can see that every arrow here is a meal. So you can see this person ate dinner at 6 p.m. The blood glucose went sky high to over 250, around 300. They injected themselves with insulin. The glucose went down. Then they went to sleep. And during the night, they did not eat. The glucose level went really down to very low levels. They ate breakfast. Glucose went really high again. Injected insulin. Glucose went down, but not down enough, etc. So you can see that this person spends a huge amount of time really above even what's considered high glucose. So that's really high percentage of time spent with high glucose in the blood. In the middle bar, you can see a person that's using a dual hormone pump. This is a pump that continuously gives the person insulin and glucagon. The main advantage is that it prevents severe hypoglycemia, which is very good. But you can see that even with this most sophisticated pump that we have now, this person still spends a lot of time in the blue area, in the high glucose area, and at least some of the time in the very dangerous range of high blood glucose. People that spend a lot of time in high blood glucose can have conditions called ketoacidosis. The blood becomes very acidic. This is not a good, that's not a good situation. On the other hand, you can see what a person that has actual normal beta cells, a healthy person, do during the day. They eat their dinner, blood glucose jumps a tiny bit and immediately goes down, hovers throughout the day on a very low. They eat breakfast here on normal range. They eat breakfast here, the blood glucose jumps a little bit, But this person, almost the person that has normal beta cells, doesn't spend almost any time in the high blood glucose range and nothing at all in the dangerous white range here. So it is clear that insulin, exogenous insulin, can treat the disease, but it can't really cure the disease. But normal beta cells can cure the disease. And because of that, people that rely on insulin exogenous insulins are more exposed later in life to complications that are caused by spending so much time in a high range of glucose in the blood. The main problem with that is that the blood becomes very thick, very viscous, and doesn't flow the way it should do. So a lot of these complications are caused by small capillaries of blood being clogged or being not enabling the thick blood, the viscous blood to go through. So you have problem in the eyes with small capillaries in the eyes, like cataracts and glaucoma. You have problems in the extremities where blood goes down but can't flow up because it's very thick. Stroke and heart attack because of small capillaries being clogged. And also problems with the kidneys, because one of the reasons, one of the things that happen in when the body has too much glucose is the kidneys are trying to filter it out. The person is trying to urinate to get rid of all the glucose through urinating. So that's actually almost always one of the first symptoms of a person with diabetes, is that you see a person that drinks a lot of water and urinates all the time in a really excessive way. That usually is a reason to get diagnosed, to get check your blood glucose to see if the person is trying to get rid of the glucose, extra glucose in the blood with the kidneys. So we know that we can now treat diabetes, but we don't know, we can't cure it with exogenous insulin. And what can actually cure the disease is a person having healthy beta cells that can sense glucose and secrete insulin in a very, very sensitive way that is much more sensitive than injecting insulin. And this is really manifested when the discovery about 20 years ago of the Edmonton, this development of the Edmonton protocol, in which people were transplanted, people with diabetes were transplanted with islets from deceased donors, from dead donors. And that you can see that actually this actually cures the disease, that weans people out of insulin, and they stay in the green range for a long time, for several years. So we know that giving the person with diabetes, healthy islets or normal islets can actually cure the disease and not just treat it. The way it works is that a donor donates the pancreas after they're dead. Islets are being taken out of it and injected into the liver. And in the liver, what these islets do is that they kind of get stuck in small capillaries where they are exposed to the bloodstream. And as I said before, one unique advantage of beta cells for cell therapy is that they don't really need to be in any certain area of the body. As long as they sense the blood glucose in the blood, the level of glucose in the blood, and they secrete insulin into the blood, they're fine. So these islets that get stuck in the liver, they're actually the same source of blood that feeds the pancreas, and they just sense the glucose in the blood, secrete insulin, and the person is completely weaned of injections of insulin and doesn't spend any time under the hypoglycemia level, doesn't spend almost no time in hyperglycemia levels, really cures the disease. What's the problem? There's several problems. One is that when we isolate islets from cadaveric donors, a lot of the beta cells die during the process. A lot of the beta cells die and the islets die when they don't get in the right places in the liver. So we need at least 2 donors for each recipient islets. That is very complicated. Think about trying to find an organ match when you want to transplant the heart. It's very, very hard. Now think about trying to find two matches at the same time. They would die at the same time and have their pancreas donated at the same time. So this is very, very rare to find two cadaveric donors that are matched. Also, these islets, because they're not usually matched, rely on severe immune suppression. So the person has to take really, really heavy immune suppression drugs for several years, which isn't good either. And finally, because these islets are kind of stuck in the liver and being exposed to liver stuff and are not completely innervated and not completely vascularized like they would be in the pancreas, it takes about 5 to 7 years for all of them to be killed, to turn over. And once this is done, the person goes back to needing external insulin. So we have a principle, a way to get islets and use them to cure diabetes, but we don't have enough of them, and they're not right for the person. So the idea that was developed about, I think, 20 years ago now, is with the discovery of human embryonic stem cells first, and then iPS cells in 2006, is that we can take embryonic stem cells or human or iPS cells that can be what challenges exist in harnessing the potential of pancreatic stem cells for therapeutic agents? Oh, Rohit, we will get there in a second. So the idea was to take stem cells, try to mimic differentiation of these cells in a dish to make insulin-secreting beta cells, and then we'll have an unlimited number of beta cells we can transplant to the patient and cure the disease, not just treat it. The problem is that making beta cells that can actually release insulin in the body is a really, really tough task. Because in contrast to organs that differentiate very early in life, like heart muscle cells or some type of neurons that you can actually easily make in a dish, making beta cells or making the right pancreatic cells is very, very hard. Because the development of the pancreas is a very complex and late process during development. So first you need to make definitive endoderm. Then you need to make a gut tube from which the pancreas becomes. You need to make the right area in the gut tube that will make pancreas and not, for example, liver or lungs. Once you got the area where the pancreas will become, you have to mimic a very complex process called pancreas branching, or the organogenesis of the pancreas, where all the acinar tissue and the islets are formed. Then you need to make those 2% of cells in the pancreas that are the islets. Where did I cursor there? That are the islets. There are the islets in the blue here. And once you have that, you can maybe try to see if they respond well enough to glucose. Just to look a little bit of how it happens. Again, the pancreas is an organ that buds from the front of the gut tube. This is the pancreas here, the orange thingy here. This is our gut tube. We need to take the cells in a dish, move them or direct them through gastrulation, and choose for differentiation of endoderm, which is the inside-most germ layer, and the last germ layer to develop through gastrulation. Then we need to specify the endoderm to foregut and select it against midgut cells and hindgut cells, unless we want to make colon or make small intestine. These two cell populations won't help us much. So we need to find ways to make these cells be foregut. And then we need to find a way to make them actually the right organ that comes from the foregut. Now, this is not only the problem of directing the cells to differentiate. There's also some morphogenesis challenges that are happening when we make islets for Langerhans. For example, to make the islet, the cells have to go through aggregation and movement in the pancreas. So in the embryonic pancreas, the endoderms or the islet of Langerhans will form from these endocrine progenitors, these brown cells in here, that actually pop out of the pancreatic duct as single cells. They don't all differentiate in the same place. They pop up as single cells. They somehow migrate through the tissue, find each other to make an islet, and go around blood vessels to actually cluster together. So we need to mimic this clustering and migration to help them differentiate. And finally, during this migration and differentiation, these progenitor cells will actually choose what type of the 5 endocrine cell types in the islet it wants to be. So we need to select the differentiation pathway that will lead us to beta cells and not, for example, to alpha cells. We'll make glucagon and secrete and help further increase our blood glucose if we transplant it. So this was a very challenging problem in what we called applied developmental biology. How do you mimic very, very complex developmental biology to make a very rare cell type that needs a lot of direction? Now, around 2005, there was a breakthrough in that in a company called ViaCyte. What they actually did in their lab in San Diego is that they took a lot of lab technicians, put them side by side in tissue culture hoods, and make them tweak and change all the signals that we knew for mouse development and human development on human embryonic stem cells, and they were able to make pancreatic endoderm and later make endocrine progenitors. And that was a really big advantage where we could actually have a stage-like protocol where we could use factors that we know for differentiation, like activin and Wnt, FGF. These are morphogens and retinoic acid that we know will help derive the cell through this process. There's a very beautiful paper in Nature Biotechnology from the lab of Jeff Millman in St. Louis from a few years ago, where he showed that he uses a mechanical, a molecule that does something to actin filaments to mimic the mechanical process of this branching morphogenesis in the islet, and makes even better use of the protocol. And from that on, a lot of labs have tried to tweak and play with this 5 stages protocol to make better and better beta cells. We now have the biggest breakthrough after that was in 2014, astonishingly 10 years ago, where 2 papers at once were able to make glucose responsive beta cells. So that was really, really good. They use actually an upscaling method where instead of growing the cells on tissue culture dishes, they grow them in 300 milliliter flasks that spin. They have a spinner that steers them all the time. And you get clusters of beta cells that really look like islets. Most of them are beta cells. The differentiation of the beta cells is not 100%. We call them stem cell-like beta cells or islet-like cells because they look like beta cells. They don't respond 100%. They need to be put in a person or in a mouse, for that extent, for about 6 weeks to 2 months until they actually mature. We don't know exactly how they do that, but it feels like they sense the fluctuation in glucose, they sense circadian rhythms, they sense nutrition, and they kind of tweak themselves to secrete the right amount of glucose in each time. And we can now, and this is from the lab of Doug Melton, where I did my postdoc. I was not involved in this research, actually. Can make billions of stem cell-derived beta cells from any person. So theoretically, we can cure type 1 diabetes. Can we or can't we? I'm asking you. If anyone wants to tell me now with billions of beta cells from a type 1 diabetic patient, have I solved the problem? Have this problem been solved and we can now transplant those into patient and cure diabetes? Who wants to guess? Anyone wants to be brave and guess?
Hello, sir.
Yes.
Yes, sir. For type 1 diabetes, if you're injecting these IPS, these islet cells, means again these immune cells will attack, no, sir?
Exactly. So that's what is your name?
Prashant, sir.
That's an excellent observation. So type 1 diabetes is a disease in which the cause of the disease is an autoimmune attack. So it doesn't help me that I can make billions of cells from the same person. If I put them back into the person, they will be attacked and killed, and I go back to disease. So the frontier of the research now in type 1 diabetes derived stem cells, we know how to make stem cells. The frontier has moved to ways that we can use them or transplant them into the body without them being rejected. Now, if you use iPS cells from a person, the immune rejection will be, there will be an immune rejection, but it won't be as severe as it is from two different people that are used from cadaveric donors. So we deal with immune rejection. It's a little bit less severe, but we still deal with it, and we have to have some solution that we can protect from it. So now their research really goes into mainly bioengineering direction, and they're very, very interesting process. What I'm going to tell you now is two different approaches that are actually used really now. One is an approach that is taken by ViaCyte, the company that first made the first differentiation. And this approach uses kind of encapsulation device. You can encapsulate the cells and put them back in the body, and they won't be rejected by the immune system. What they do here is they take the beta cells. This is the beta cell differentiation that they have, with insulin secreting beta cells in blue, glucagon in red, and somatostatin in green. So they actually make all cells of the pancreas. They don't make it in the right percentage, but the percentage kind of being corrected in the body. And then they put it into this device. The device is about the size of a tea bag. It actually looks like a tea bag. And they put it in the back of the lower back of the person. And the device has a membrane that should allow glucose and oxygen and other nutrients to get into the membrane. It allows insulin to go out of the membrane, but its pores are small enough that immune cells cannot get in. And that's been a clinical trial, really big clinical trial that ended about a year ago. And it looked very promising at the beginning. So they transplanted that. They transplant endoderm. So they don't transplant actual beta cell. They transplant the progenitors and let the progenitors finish differentiation in the person. Here you can see how it looks at the beginning with vascularization around it. And at the beginning, it looked like these procedures that can rescue patients, but the conclusion of this clinical trial that happened about, I think, a year ago was a huge failure. And the reason for the failure was that it wasn't appreciated what the immune cells will do when they can get in. So what happens here is the immune cells sense there's something wrong here. They sense that these are beta cells and they want to kill it because they're autoimmune. There's autoimmunity against beta cells in this person. These were not actually iPS cells from the same person, but iPS cells from other people. We don't have personalized availability of those yet. But the immune cells could not get in, and they did what immune cells do when they can get in somewhere. They build a scar around it. So they build fibrosis, and what happens in pretty advanced stage is this membrane was completely clogged by scar tissue, by fibrosis around it. nothing could get in, nothing get out, and all the cells die. And this was a huge failure. ViaCyte is now being bought by another company called Vertex, and they are using some other really cool devices to try to block this. They're actually a really, really interesting mechanical thing that I can't really explain, where they try to use similar membrane, but not in the shape of a tea bag, not flat, but tiny, tiny bubbles in which about the size of an islet each, and some mechanical angles of this that I can't really explain prevents scar tissue to be built around them. And that's now I've seen that about a year ago being tried in mice, and I think there's a try in monkeys. And if the try in monkeys will work, they're probably going to continue to go to human. Another approach is being taken by a company called Vertex. They now bought ViaCyte. So actually, Vertex is the only company that is doing that on high scale. And they took another approach. And what they did is they said, okay, so we solved the problem of not having enough beta cells. We can make billions of beta-like cells in a dish. What we do is we're just going to transplant them into the liver the way we do with deceased donor islets, with cadaveric islets, and we just flood the patient with immunosuppressant, and we see if it works. This is from 2 years ago when they started the clinical trial. This is the first person that got the islets, the stem cell-derived islets, into the liver on immunosuppressant, that you can see here in this New York Times article, that is very happy. Because at that time it was thought that I think it's still, that he, after 90 days, is completely cured from diabetes. His C-peptide, his connective peptide that is being used to sense human insulin, went from undetectable to a very measurable levels. His daily dose of insulin went down from 34 units of insulin a day to 2 point something. So he really is almost weaned of insulin. I think later he was shown to be completely used of insulin, completely weaned or completely unrelied on insulin. It does not need insulin at all. So that was the big success. Unfortunately, when I made this presentation for your class, I looked back at what happens with this trial, and I saw that it was stopped because just a month ago, 2 months ago, the trial was stopped because 2 patients died. Now, trials of that sort are stopped when a person dies, not only because they die of the device. So someone can die of a heart attack, someone can die of a car accident. If a patient dies on a clinical trials at that level, everything is being stopped. So we don't know if this person actually died because of the disease. I doubt that they did. If anything, maybe the immunosuppressant or something unrelated. But I think everyone's waiting for this to continue again, to get approval for the trial to continue and see whether they can actually cure diabetes. Now, if you have any questions for now, before I spend the next few minutes on stem cell in disease modeling, I'm happy to answer them.
Barak,
Yeah.
just wanted to remind you, maybe about 10 minutes we have altogether.
Yes.
Is that good enough for you?
Yes, I can actually. I don't have to do the disease modeling stuff.
You should, because it's an opportunity for all of us to listen to. Come on. Yeah.
Okay, I will. I have 7 more slides. I'll give them, and then I'll move on. I'll only do type 1 diabetes. So Rohit asked me what advancement or development have been made by ViaCyte in recent years regard to the use of preferential diabetes. So I think ViaCyte, that's a good question. ViaCyte now are working mainly on mainly on delivering. So they're working on their encapsulation device. And I haven't seen much progress with it, but I mean, we're looking, and these are all proprietary discoveries now when it's in the hand of companies. So we don't see much until something actually succeeds. So I'm looking forward to see what Viasat is doing. Now Viasat are being bought by Vertex. Vertex now is they bought Doug Melton's company, they bought Viasat, they bought everything, and they're pretty much the only huge company that is doing that, which I don't think is a good thing. So stem cell, yeah, another question. Thank you, sir.
I'll take up this question now. Actually, let's finish this because we are hitting about 10 o'clock. We'll be getting off the schedule. Okay.
Okay. So stem cell derived beta cell in disease and disease modeling. So we can understand diabetes, type 1 diabetes. And our understanding of type 1 diabetes in person is very, very limited. The reason is that most of the disease happening, most of the things that happen in person happen before the disease is diagnosed. So you say you have a genetic risk, you have the beginning of an immune attack, and you have silent beta cell loss. The immune system starts killing the beta cells and killing the beta cells. And only when enough beta cells are dying or have died, the person is usually diagnosed, and everything that happens before is a black box to us. We don't know that. So we do have some. So there are several tissues involved also, not just the beta cells. There is the islet where the beta cells are being killed. There is the immune system that we don't know what's going on there, and of course the thymus and the bone marrow that are also related to immune building. So we have one mouse model for autoimmune diabetes that has been used for many, many years. It's called the NOD mouse. This NOD mouse has spontaneously developed autoimmune diabetes. The picture that I showed you before, actually from NOD mice. It showed many symptomatic phenotypes with human type 1 diabetes, but it's a genetic model that represents only one model of type 2 diabetes. So type 1 diabetes. So there's a lot of questions that we don't know and we cannot answer with this NOD mice. Although it answers a lot of questions, we still don't know, for example, if the pancreas or the immune system are responsible in all patients. Maybe it's different in different patients. When does it actually start? How does it actually start? So what is the first antibodies? We don't know them. When we see the disease, usually the antibodies are against, the autoantibodies are against the most common genes, with usually insulin. Does it always start the same way? So can a person with the same susceptibility have the disease starting a different way? And is human type 2 diabetes happen to the mouse? Yeah. So recently, the lab of Doug Melton and actually other labs have been able to generate iPS cells from people with genetic predisposition to type 1 diabetes, and they're using them in some really cool ways. One way that I want to mention here, that's my last slide, is the way to model how type 1 diabetes develops in different people by using humanized mouse. What you can do here is you can take a mouse, you can irradiate the mouse, you kill all the immune system, and you kill all the beta cells, you take the beta cells out, and then you make iPS cells from people with type 1 diabetes. You make immune cells from the same person and beta cells from the same person. And then you take those immune cells and beta cells that represent one person, and say you put them in 100 mice, and you follow these mice. And what you can then say is whether the disease happens always at the same stage, whether the first antibody is always insulin, or is it something random? And a lot of these questions, I think, we're going to see in recent years when labs are starting to use diabetes or iPS cells to model the diabetes. There's a lot of other things we can do. We can play around with CRISPR and edit genes in the immune system and leave them in the beta cells, edit them in the beta cells and leave them in the immune system, et cetera, et cetera. And we see how that goes. So I think academic research is going to be a lot about modeling diseases with stem cells. I will skip modeling type 2 diabetes. And I will only say that we can now use organs on a chip. So we can use iPS cells to mimic or differentiate many different tissues. So we can make fat cells and islets and liver cells and see where insulin resistance actually start in a dish and mimic that. We can make gut cells. We can even infect the gut cells that we make from iPS cells with different types of microbiome and put this on a chip and see how a stem cell derived islet responds in different genetic predisposition to type 2 diabetes. And with this, I will end. I'll just recap that we talked about the endocrine pancreas, what are beta cells, how they function in normal glucose homeostasis. We talked about how they go wrong in diabetes mellitus. We talked a little bit about approaches to use iPS and human embryonic stem cells to make new beta cells. What are the challenges now that we've managed to make them? What are the challenges in the delivery? And a little bit, tiny bit about stem cell-derived beta cells, disease modeling, and drug discovery for diabetes. And with that, I will end.
Well, thank you, Barak. You can take up this question which was in the chat box. You can have a look at that. What was there any specific?
No, actually, I think I answered that.
Okay, good. So
yeah.
you have paid attention, and I think I have shared the information with you. There is a Majid Ibrahimi from Australia. He talked about, he actually he's a guy from Iran, and then he did his PhD from Singapore, did a postdoc at Harvard, and then moved as he's an engineer, by the way, not a biologist. So he is now making a lot of these kind of mechanical things. And he has given delivered a talk on the, I think, second or third day in our workshop. You can have a look at that, and his contact is there. And it's a potential collaboration for as far as the microfluidics and the organ on chip are concerned. Okay,
Thank you very much. I'll take a look. Yeah, I think this is where most of the science is going.
Okay.
My research actually is more in how islets develop and what happens to them in diabetes. And we're trying to find out why the cells organize the way they are. So we're working on how the cells find the way to the islet during development and what happens to them in diabetes.
Yeah. If any
mainly in type 2 diabetes.
Okay, very nice, very nice. If any of amongst us, among the participant has got any question?
Don't be shy.
Yeah. Prashant again has got a question for you. Prashant is from my lab. Okay. Yeah.
Hi, sir. How long it will take to produce this beta cells from iPSCs or any source of pluripotent stem cells?
Yeah, this is really long protocol. It's a good question. It's a very long protocol. It takes 37 weeks to make cells that make 37 days, sorry, to make cells that actually make insulin. And then it takes them about 2 months in a dish until they can actually mature.
So
6 weeks, 2 months. It's a very, very long protocol.
So if we can produce within like 3 months, 4 months, means we can go for patient-specific beta cells instead of using this drug delivery system. We don't know how this immune system will react. We can go for the patient-specific beta cells development.
Yes, I think we can. One of the things is that every cell type, every cell line is a little bit different in the way it responds to differentiation. So every time you make a new cell line, you actually have to tweak the protocol a little bit until you get the right differentiation. We are actually getting this problem now. We have a patient with a genetic disease that we're trying to differentiate. The beta cell has been working on it for 2 years, and we're not getting we're getting some beta cells. We're not getting satisfactory differentiation because every cell line responds a little bit different to differentiation protocols.
And why we have to inject it into the portal vein like vascular system? If you're injecting bulk of cells, means it will block the vascular system. We can inject into the pancreas itself.
So that's actually a really good question. The pancreas, the problem with the pancreas is that if you hear any surgeons ever, the first thing they'll tell you is don't touch the pancreas, don't mess with the pancreas. The reason is the pancreas is full of digestive enzymes. And once you start injuring the pancreas, it starts eating itself digesting itself with trypsin and chymotrypsin and everything. And this is a terrible condition. So surgeons try as much as they can not to inject anything and transplant anything into the pancreas. It's a really
Okay.
good question.
Thank you.
Thank you, Barak. And it's wonderful to have you over here, and I'll be in touch. Yeah, good to see you, and say hello to everyone in the family. Okay.
Thank you, Sujoy. Very nice seeing everyone. All right. Good day.